Auzu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. So today we are going to study about the salmonella. Salmonella. So what are salmonella? What are its characteristic features? So salmonella, it is a gram-negative rods having a straight shape. Having a straight shape. Second one is that it belongs to a group known as Enterobacteriaceae. So thus it contains all the characteristics of Enterobacteriaceae, like the one that it is a facultative anaerobe we have already discussed in the Escherichia coli. It converts nitrites to nitrates for the purpose of energy. It also ferments glucose and it is oxidase negative. Although it ferments glucose, but it is a non-lactose fermenter. In contrast to the E. coli, which are lactose fermenters, and this property will help us in the lab diagnosis. The fourth property of the Salmonella genera is that it produces H2S. H2S, and it also helps in the formation, or it also helps in the diagnosis of Salmonella in the lab. It is citrate positive, meaning that it uses a citrate for the purpose of energy, in contrast to the Escherichia coli, which uses the acetate. Now, this Salmonella is classified clinically on the basis of clinical signs and symptoms into two groups, or into two species, two types of species: a typhoidal species and the non-typhoidal species. The typhoidal species cause the typhoid fever or enteric typhoid fever or the enteric fever, and the non-typhoidal species causes the diseases like gastroenteritis and the septicemia, etc. The typhoidal species include the species like the one Salmonella typhi. The second one is the Salmonella paratyphi. Salmonella typhi and Salmonella paratyphi. And the non-typhoidal species include the species like the Salmonella fulveraceus, and the second one is the Salmonella enteritidis. Now let's talk about the antigen present in the Salmonella. So it consists, like Escherichia coli, three antigens. The first one is the O or cell wall antigen. The second one is the H or flagellar antigen, say Escherichia coli, and the third one is the K or capsular or VI, i.e. virulence antigen, because it is the main virulent vector for this Salmonella. So what does this O or cell wall antigen helps in? It is used to subdivide the Salmonella into many groups, like from the group A to I. It is used to subdivide into many subgroups. The H or flagellar antigen has two forms. It may be phase 1 in the phase 1 form, or it may be in the phase 2 form. And only one is present at a time, not both. In the K or capsular or the virulence antigen, it is the main antigen, and it is antiphagocytic, and it is also used for the serotyping. Serotyping. It is antiphagocytic and hence it helps in the pathogenesis of the typhoid fever or other diseases. Now, coming towards the epidemiology, of the Salmonella. So the main important organism in the Salmonella is the Salmonella typhi. Salmonella typhi. And this Salmonella typhi is only present in the humans. There is no other reservoir except human, and only human to human transmission occurs. And it is mainly through the fecal-oral route. Fecal-oral route. The other species like the Salmonella oleracea or enteritidis or the paratyphi, they can be found in the eggs, in the poultry, undercooked meat, dogs, and other pets in addition to humans. Although they are also present in the human, but they can also be found in such kind of things in contrast to the Salmonella typhi, which are only present in the humans. Now, after studying the epidemiology, let's study about the diseases which are caused by the Salmonella genera. So, in the disease, we will study about the causative agent. The second one, we will study about the pathogenesis behind which they causes the disease. And the third one, their clinical findings. So the Salmonella species causes 3 types of diseases. The first one is the typhoid fever, the most important. The second one is the gastroenteritis, and the third one is the septicemia. We will first study about the typhoid fever. So, typhoid fever. It is one of the most important disease caused by the Salmonella species, and they are mostly caused by 2 species of the Salmonella genera. The first one is the Salmonella typhi, and the second one is the Salmonella paratyphi, in which includes all the 3 agents, the A, B, and C. All the A, B, C paratyphi cause the typhoid fever, but the most important is the Salmonella typhi, which causes this typhoid fever, also known as the enteric fever. Now, what's the pathogenesis behind the disease caused by the Salmonella typhi and the paratyphi? So, the first step in their diseases causing pathogenesis is they enter the stomach with the help of food and water contaminated with such kind of bacteria. When they enter the stomach, it passes through the stomach into the small intestine. In the small intestine, what they do in the small intestine, they enter the phagocytes of the pair pages present in the submucosa of the small intestine. What are pair pages? These are the lymphoid tissues, pair pages. These are the lymphoid tissues present in the small intestine, and they helps protecting our small intestine from different kind of diseases. So they enter the pair pages, the phagocytes of the pair pages, but the important point to keep in mind is that they are not killed by the phagocytes. And it is because that they contain a special type of capsule called the Werlen's capsule, and this Werlen's capsule, as we have already discussed in the antigen, is antiphagocytic. And this antiphagocytic property of the Werlen's capsule will helps the Salmonella typhi against the killing effect of the lysosomes and phagosomes present in the lymphocytes. Now, from these lymphocytes, the lymph ducts passes into the bloodstream, and with the help of these slim ducts, they are passed directly into the bloodstream to cause the bacteremia. It is the stage where some clinical findings in the first week appears. From through this bloodstream, then this bacteria passes into different organs like the skin, the bone marrow, the spleen, the gallbladder, the liver, and the kidneys. And the most important of these organ is the gallbladder, because when it enters the gallbladder, there is establishment of the carrier state. And this carrier states appear in about 3% of the patients, and it is mostly present in the women having already a gallbladder disease like the gallstone, etc. They are mostly present in the women, and it accounts about the 3% of the patient. And from this gallbladder, it is excreted into the intestine and then into the via the feces into the external surrounding via the gallbladder. And this will help in the transmission of the bacteria to other organisms, as we have earlier discussed in the epidemiology, through fecal-oral route. So this gallbladder plays a very important role in the establishment of the carrier state of the pathogenesis. And keep in mind that when it spread to the bloodstream to this kind of organs like the skin, bone marrow, they mainly reside in the reticoendothelial cells of these organs. And after the death of these reticoendothelial cells by these bacteria, they will invade these organs and will cause the characteristic signs and symptoms of the disease. Now, as we've already discussed the pathogenesis, so now let's study about the clinical findings. And the clinical findings of the typhoid fever or the enteric fever mainly depend upon the duration of illness. I.e., in the first week, we will discuss in the first week. In the second week, what will be the sign and symptom, and in the third week, what will be the sign and symptoms. So in the first week, there will be the slow onset step ladder pattern of fever. The first symptom will be the slow onset of slow and step ladder pattern of fever. Step ladder pattern means that they will increase in a step ladder pattern like this, not acutely like the malaria, etc. There will be slow and step ladder pattern of fever. There will be constipation. Diarrhea may occur in the early stage but then resides. There will be vomiting, but they will be much less, and there will be malaise. These are general signs and symptoms caused by the Salmonella typhi. And the important one in the first week is that there will be appearance of red rose like spots, which is a type of rash on the upper abdomen, mainly on the upper abdomen. If it is left untreated, then it can lead to the second week, and here the bacteremia is much more, because the phagocytes in the reticoendothelial cells die, and there will be much more release of the Salmonella typhi and paratyphi. So in this stage, the bacteremia becomes sustained, and it is characterized by the signs and symptoms like there will be high fever, tachycardia, there will be a tender abdomen, tender abdomen. There will be hepatosplenomegaly, and thus there will be the abnormal LFTs, liver function tests. And also we can see leukopenia and anemia. Now, if it is left untreated, it will progress into the third week, and the third week is mainly characterized by the signs and symptoms like the toxemia, there will be delirium, disorientation of the patient, and it can also lead to the coma. So they are mainly CNS related, and if it is usually the disease resolves in the third week, but if it is not resolves, or if it is rated, if it is untreated, if it is untreated, then it can lead to severe complications like the intestinal hemorrhage or intestinal perforations. So if it is left untreated in the third week, then it can lead to complications like the intestinal hemorrhage and the intestinal perforations. So these were some clinical findings, the pathogenesis and the disease and the causative agent of the typhoid fever.